So welcome to our first virtual session for biology. I'm going to go ahead and share my screen. And we are going to be talking about the properties of water and variables. Right, so properties of water was probably, or is probably, the most important piece of the module. Kendrick, no. Down. I trained my dog, so I have to practice with him, but now he knows how to stay down for extended periods of time. It's really exciting. It's still really hard for him, obviously. He's crazy. Okay, so properties of water is the most important part of this module, I would say. The rest of it is good, but not as useful, and variables are super important, right? Some of you may have already checked out the collaboration project. Most of them are involving some pretty intense experiments, right? So a foundation of variables is super important. So rules for our virtual sessions, which everyone so far is following: camera on, right? You don't have to have your face like directly in the camera. You can point the camera down or up a little bit if it makes you uncomfortable. Participate, right? So we're gonna do a lot of participating in the chat. So feel free to type in the chat. If you'd like to speak instead, you're also welcome to do that. I will probably mute you occasionally, not to make you be quiet, but because then there's tons of weird noises going on. And then be respectful, right? Respect other people's opinions, respect me. Don't foresee any issues with that, but keep it in mind. So we're gonna start off with a little warm-up question, which I know Elizabeth is gonna like. Elizabeth emailed me yesterday asking about the Earth's early atmosphere assignment, which is top 5 most obnoxious assignments that you will see in the class. So this question kind of relates to that. What does it mean to have a universal code, and why is that important? So I want you guys just to we'll spend about a minute thinking about this, and if you would like to suggest an answer, feel free to type in the chat, or once our minute is up, you may speak. So think about what that means, a universal code, and why that might be important. Go boy. Okay, so what do you guys think? What does that mean, a universal code? Okay. Isabella says, a universal code is all organisms having the same DNA sequence. I'm going to qualify that slightly, right? Really meaning that they all have the same letters in their DNA or the same molecules, right? We don't have exactly identical sequences, but you are right that they have some of the sequence in common, right? Even we have 40% of our DNA in common with a fruit fly. If you look at a fruit fly, no way are you thinking, oh yeah, I'm related to that fruit fly, right? But you are. 40% of your DNA is the same. So same with chimpanzees, right? 98% of your DNA is the same. That 2% clearly makes a big difference, right? Because you don't act and look like a chimpanzee. And you're also right in your second point here that it supports the RNA world theory, right? So there's all these theories of how life started. And there all of them have evidence to support them. There's some theories that life started in the deep vents in the ocean where there was warm air and a bunch of weird stuff mixing together. There's an RNA theory. RNA is super volatile. It can break down and build itself really quickly. So there's a lot of thought that maybe RNA had to do with the beginning of life. There's also my personal favorite theory, panspermia, which is that life came from outer space on a meteor or some kind of fragment from space, hit Earth, and then that was the beginning of life. Right? So there's all these different theories, and a universal genetic code really does kind of fit with all of them. But essentially, all living organisms have the same molecules in their DNA. Right? And we represent them as 4 letters when we write out a DNA sequence. But that doesn't necessarily represent what it really looks like. We're going to learn a ton more about DNA in the future. So if this assignment seemed to go over your head a little bit, that's fine. Once we've learned about DNA and RNA and proteins, we're going to be like, oh, I see what they're trying to do there. So if we have time, we'll come back to that assignment at the end as well. And if you want additional help with that, we can talk about it. So today we are going to define and describe the major properties of water, define the major types of variables in a controlled experiment, and then we're going to practice identifying variables in some experiments about water. So we are going to be using Cornell notes for this class. Type 1 in the chat if you have used Cornell notes before, or you've heard of them, maybe they've been suggested to you. Okay, so Elizabeth and Isabella have both used them. Let me see. I'm waiting on, I don't know, I can't see what your name is in the, when it's on the video. How do I fix that? Let's see. No, not yet. Okay, Harrison, there we go. Okay, thank you. So, oops. So we are going to use Cornell notes for this class, and once per module, at the end of the module, I'm going to ask to see those notes. Just one lesson of notes. I will be asking about properties of water notes for this module. So if you're here and you're taking notes with me today, then you should be ready to have your notes to turn in for that. Now, there's a ton of resources about Cornell notes. Today, we're just going to focus on using the template and getting all the main parts. And in these virtual sessions, I will be kind of helping you through the process. So this doc that I just sent in the chat, that is the Cornell notes template. So what it looks like is this. And you can either write on paper or you can write in the document, right? You'll have to make a copy of it for yourself. That's up to you. I know some people prefer to write by hand, totally fine. So what you're going to be doing is you're going to be filling in the topic or the objective, right? I just switched from the objective slides, but you could say properties of water, your name, your class period, and the date. And I will tell you what the essential question is, and I'm also going to tell you some of the questions you could put on the side and the notes content that you should be putting. You will write your own summary at the end. Right, and if you're really familiar with Cornell notes, you might even go back and do the next steps, circling and underlining, right, crossing out information that wasn't important to you, etc. But for now, we're going to just start off by getting something down on the paper. So if you would like to use the template digitally, just take a second right now to make a copy of it and get it set up for today. If you already have notes about the properties of water, open those or get those out so that you can add to them, because I'm not going to make you write stuff that you already wrote. So if you're new to Cornell notes, you might also want to come back after this session. There's a video here that Mr. Ling made, and there's also a PowerPoint that I made for Avid showing like step by step how to do the notes. So if you're like, I have no idea what these are, it might be helpful to you to either watch that video or look at the PowerPoint, kind of get a better concept of what they are. But we're not going to spend time watching the video during this. So, our essential question is, why is water such a special substance? Very, very thought-provoking question, I know. We really probably don't think of water as being that special, right? Because we're so used to seeing it, used to interacting with it. You're like, water seems pretty normal to me. Well, it's not. So make sure you have this essential question written down in the part of the notes that says essential question. If you had your own essential question, like what are the properties of water or something like that, that's also fine. So what is water? Here is an example where what is water is going to be the question that you're going to write on the left-hand side. I already kind of did that part for you, coming up with the side questions. And our actual notes here that are going to go on the right side are about water. I'm going to give you 30 seconds to either type or write, and then we're going to start talking about what this stuff means. So you're going to type this in. You should have, actually, let me make a copy of this so I can write in it. Ignore this random, slightly more negative arrow. That will come back in a second. Yeah, so your essential question is going to be, why is water such a special substance? And then over here, your first side question, oops, should be, what is water? And in the notes part, you should be writing what you see here. H2O, water is polar, water is a universal solvent, etc. I'll give you guys about 20 more seconds. Okay, so what is water, right? H2O, that's literally the formula. Usually the 2 would be a subscript, right? But you could also write it like this. Water is polar. Does anyone remember or maybe learned what polar meant if they went through the mini lesson about properties of water or the regular lesson? What does it mean for something to be polar? Anyone have any ideas about polar? Well, it has the word pole in it, right? Really, polar means that there's 2 sides to something. So in this case, there's a positive side and a negative side. There's 2 charges going on here. When you take chemistry, you'll learn more about polarity of molecules and all that kind of stuff, but water is a polar molecule. right? So if we look at the picture of water over here, it does look like a Mickey Mouse. We have our 2 hydrogens and our oxygen. We have all our electrons here drawn. The oxygen electrons are in red and the hydrogen in gray. Oxygen is a way bigger molecule, and it's also mean. It steals electrons, right, or it tries to. Hydrogen won't give them up, though, but it doesn't share equally. So oxygen keeps the electrons more than the hydrogen, right? And electrons have a negative charge. So that means that the oxygen side is actually more negative, and the hydrogen side is more positive. So that means that water molecules are actually attracted to each other like magnets, right? If you think about a magnet, there's a positive and negative end. If you have the positive and negative next to each other, they quickly go together, right? Water is very similar. It's the positive and the negatives are actually attracted. So water will want to be next to each other, more so than other substances, right? So that bonding here between the hydrogen and oxygen creates this uneven distribution of the electrons, and that's how we get a polar molecule, where the oxygen is actually a little bit more negative. So water is also a universal solvent, right? It's called that because water can dissolve more substances than any other substance on Earth. So you might even think about all the different things that can get dissolved in water, right? Sugar, salt, etc. Just for us, like for food and beverage purposes. But other things can get dissolved in water as well, including like pollution and pollutants. So water can dissolve almost anything, and a lot of it has to do with this polarity. Those positive and negative charges often attract other substances as well. Another weird thing about water, it exists as a solid, liquid, and a gas on Earth, right? A few other substances can, but most substances may exist as a liquid and a gas, but not a solid, right? Because it needs to be like negative 300 degrees for them to be a solid. Water in a normal temperature situation can exist as all three, right? In fact, on a nice, cold, icy day, which hopefully we won't have too many of those this year, you are experiencing all three phases at once. If there's ice, that's a solid. If some of the ice is melting, you have a liquid. And then there's also water vapor in the air, right? So on a winter day, you could be experiencing all three phases at the same time. And another weird thing about water, the solid is less dense than a liquid. Usually solids are more dense, right? Matter gets more compacted. Usually that means solids are heavier, denser. Like if you think about a substance like, I'm trying to think of something else that could be a solid and a liquid that we might see on Earth. There's not really that many of them. Metals, for example. So think about if we took like a watch, right? And it's made out of metal. It's a nice solid, it's very dense. If you melt it, right, and it's a liquid, it's gonna be, the solid would still sink in the liquid, right? The solid is still more dense than the liquid. Whereas with water, the ice actually floats on top of the water, which is also a big deal. So, sorry you're late. Well, we're glad you're here, Destiny. We're taking Cornell notes, by the way. Okay, so that was my little spiel about what is water. So, oops, next part, I'm like not used to using Zoom anymore. Too many clickings. All right, is what specific properties does water have? Oops, forgot to erase that one. So these are vocabulary terms here that you should have gotten from the properties of water lesson, right? If you didn't and you haven't written them down, this would be the time to do so. By the way, this whole PowerPoint is going to be posted. So if you missed something or I went too quickly through something, you can always go back and look at it again. So we have adhesion, cohesion, surface tension, and capillary action. So adhesion means water will stick to other items, right? Because of that polarity, water's attracted to other substances. Cohesion, you might think like like cooperate, collaborate. Water sticks to itself, right? Water likes itself too. It wants to be next to each other. Surface tension. Water can support a certain amount of weight on its surface, right? So you might have experienced this if you've ever seen like an insect walking on water, right? Certain things can float on water because of surface tension. You might even have seen this if you've ever filled up a glass of water to the very, very top. You'll notice that water makes a dome shape, right? It doesn't immediately spill. It kind of makes this bubble, and then eventually it will spill. And capillary action is the property in which water will move against gravity. So if you have a tube, water can actually move up the tube against gravity, right? That might not seem that interesting, but really that's how plants can exist. Very important for them. So I'm going to go on. If you want to see these words again, I will show you where the PowerPoint is at the end so that you can access it. So here's some pictures of those exact things. Hold on a second. Kendrick, come. Down. Good boy. Okay, so we have a picture here of water pooling on a leaf, right? You might have seen something like this. Water can do that on clothes even sometimes. So this would be a good example of both adhesion and cohesion. The water is attracted to the leaf, right? But it's also pooling in these little bubbles of water. Because water is cohesive, it's also attracted to itself. Here we see a lovely stick bug walking on water, right? You can see the surface tension here as the bug presses down on the water, right? That's the water actually resisting that force. And then this picture here with the red capillary tubes here is a demonstration basically of if you have a thinner tube, the water will go up higher. But basically, water will move up that tube without you having to do anything, which is pretty cool. It's because the water is attracted to the side of the tube and it's attracted to each other, so it will start climbing up the tube. So like I said, that's super important for plants, right? Think about a tree. A tree is 50 feet tall, maybe, possibly taller. They have to get water from the ground to the leaves. Not an easy task, especially since they don't have like a pump, right? Capillary action is actually can pull water all the way up to the top of a tree, which is pretty cool. So, what is going on with my computer here? Uh-oh. Okay, there we go. So why should we care about this for the real world? Right? A lot of you guys mentioned some of this stuff in your Properties of Water lab, making those real world connections. So water dissolves substances, which means it can dissolve things in a good way, right? Salt. It could also dissolve pollution, not so great. Minerals and rocks, right? Water is super important in actually weathering and breaking down rock. And a lot of that rock can get dissolved in water. Right? Oxygen can dissolve in water. Carbon dioxide can dissolve in water. So all sorts of things can dissolve. Ice floats on water, right? This allows marine life to thrive even in the winter. So even when a lake is completely frozen, underneath the water is not frozen and fish can still live, which is pretty cool. And then capillary action allows plants to grow tall, which I already said because I am just so amazed that that actually works. Okay, switching up here. Let me know if I'm going too fast, guys, by the way. If you're like trying to write something down and you're like, it's much harder for me when I'm not actually writing how quickly I should be going here. So what are variables, right? This is kind of a review of, hopefully you've learned about variables in every other science class that you've ever taken. But this is the way that I like to remember what they are. So we have our independent variable, our dependent variable, our control variables, and our control group. Our independent variable is what the scientist is choosing to change, right? So when you're designing your experiment, what conscious choice are you making? What are you making different so that you can see if it matters? The dependent variable is what you're measuring. So what are you measuring in your data? Control variables are what you're keeping the same, right? So your independent variable should be one thing that you're changing. Everything else should be the same. These are hard. Also notice that these are variables. Ooh, what is this thing? Ooh, cool. Variables with an S, right? Because there's more than one of them. There could be 100 of them. And then we have our control group, which is confusing because it sounds like a control variable. This is our normal group, which I put normal in quotations, right? Because really, you're deciding what the normal group is, but it's what you're comparing to. There's not always a control group, right? A lot of times people get hung up on that. They're like, oh no, what's my control group? You might not have one. You might not have a normal situation that you're comparing to, and that's fine. You don't need a control group to have a good experiment. So the reason that we're going to go over this now and practice this variable stuff is because you did your properties of water lab. We're going to be doing a lab next week called Whose Doorknob is the Dirtiest, where we will be using these variables again. And then when you do your own collaboration project, you'll be using these variables. So we're going to practice together on this one, identifying these variables. So Cynthia is studying how temperature affects the amount of sugar that dissolves in water. I guess I should put an S on that dissolve, but. So what is our independent variable here? What is Cynthia, as a scientist, changing in her experiment? The temperature? Yes, awesome, right? She's saying literally how does temperature affect the amount of sugar? So she's going to be changing the temperature. She might have cold, warm, hot, right? She might have a whole bunch of different ranges. We don't know. She didn't explain, but something like that. So you are right. She is changing the temperature. What is she measuring? The amount of sugar? Yeah. So she's measuring the amount of sugar. So what would she need to keep the same here if she was really going to do this experiment, right? And even though we don't know every detail about what she's going to do, so it might be a little hard to describe these, but what kinds of stuff should we keep the same? The amount of sugar? Good. Right? We can't say, okay, well, in hot water, I'm going to put 30 grams of sugar, but in cold water, I'm only going to put 1 gram, right? Because then that's not going to tell us anything. And the amount of water? Good. The thermometer? Good. So the equipment, we'll just say, because that's going to encompass even the cups, right? Ideally, you should be using the same cup, the same thermometer, the same room temperature, the same amount of light, right? Even though those things probably aren't going to impact this, in a real controlled experiment, everything is the same except for the temperature of the water. And then what about a control group here? Is there a control group? Do we know? Is there like a normal situation where water dissolves sugar? What do you guys think? Control group, yes or no? No, you guys are right. There's not one, right? There is no normal situation with sugar and water, right? We will see an example here where there is one. But you guys are right. There's no control group. So we can see kind of how this experiment could be something that you could easily set up, right? Personally, I think the hardest part sometimes is identifying what the variables are in an experiment, especially when it's not your experiment and you're looking at what someone else is doing. Okay, so we're going to practice one more. Okay, so John is designing an experiment to see if the type of salt affects the melting of snow. He is using 3 types of salt and putting 40 kilograms of salt on top of a square meter of snow. So what do we think our IV or independent variable is here? What is John changing as the scientist here? Awesome. Types of salt. Now you may be thinking in your mind, types of salt. Right, there are types of salt. So if you remember last year in our lovely winter of snow, people were putting some salt out in certain spots, right? That is not the same kind of salt that you put on your food. You probably noticed in some cases it's like neon blue, for example, right? That's a dye that they put in there. But there are a bunch of different kinds of salt, right? So you could have table salt. There's things called road salt. There's like, which is usually calcium chloride. You could also have magnesium chloride. You could have, which is Epsom salt, right? If anybody takes Epsom salt baths, one of my favorite things. But yeah, types of salt. All right, and then what is he measuring? Yeah, the melting of the snow. Now, let me ask you guys this. How could we measure that? If we were really going to do this experiment, how could we measure how much snow melts? There's not really a right answer here, but how do you guys think we could do that? If we had a little square space of snow and we're putting salt on it, how can we measure how much actually melts? The weight? That's a really interesting idea, Jordan. So liquid water is going to weigh more than frozen snow, right? So we could actually measure the weight over time and see how much has melted. If it's in a container, we could see the ice part floating on the water, right? So you could actually measure the volume, I guess, of the water. True. Right? You can even do something as simple as sticking a ruler in and measuring the height of the snow and how much it goes down. Right? If it starts out at 10 inches and the next day it's at 9, then you can assume that like 1 inch of snow has melted. So there's a whole bunch of different ways that we could do this experiment. And I've actually done an experiment like this before, where we did road salt, table salt, and charcoal. And there wasn't really that much of a difference, but the charcoal actually prevented the snow from melting. But the table salt and road salt both melted it pretty well. Okay, and then control variables here. What would John need to keep the same? Yeah, the amount of salt, the amount of snow. Excellent. Destiny gave specific numbers here, 10 kilograms of each, or 40 kilograms of each. And for some reason I have a spot on my computer right there. Here we go. And a square meter of snow. The equipment, again, excellent, Jordan, right? Whatever he's measuring with, we want to make sure it's the same stuff every time. And then we assume here that the location is the same, right? But we would want to make sure that it is. He's not doing, some measuring at school and some at home, for example, because that could actually be different. Yeah, and the 3 types that he's specifically using, right? They should be from the same bag even. Very, very detailed, specific stuff, but it can make a difference. Now, what about a control group? Is there a normal type of salt that we use to melt snow? I have a no and a yes. There is, technically. There is road salt, right? It's usually calcium chloride, which is generally this type of salt used to melt snow. Now, this could be a control group, right? Where you say, well, usually we use calcium chloride, so we're going to see if it's the most effective. Or if you're not using calcium chloride, right, because he doesn't say he's using that, then you might not have a control group here. He might be using table salt, Epsom salt, and another type of salt that's not road salt. So in that case, right, there might be a control group here, there might not. Really, it wouldn't be super important except for in your conclusion. You would want to mention like, well, road salt maybe wasn't the best salt, so why are we using it as road salt, right? It is the best though, because they've done testing on it, that's why it's road salt. So that's an example of where there kind of is a normal situation. You might not know, right, especially because I gave you 2 sentences about this, but if you had researched to actually do this experiment yourself, you would have found out that there is a control salt. Okay, so that was it. Notice that I brought in properties of water into those experiments. So basically what I want you guys to do now is at the end of all of your lessons, all of your notes that you take, you are gonna wanna summarize the lesson. And I'm gonna challenge you today to actually summarize in 20 words. Exactly 20 words. So I'm gonna give you about 2 minutes. If you came in a little bit late to the lesson, summarize what you know. Yeah, within 20. If you could do it in 10 words, that's awesome. But the point here is you don't want your summary to be 17 sentences long, right? It should be very short, so when you read it, you're like, oh yeah, that's what that lesson was about. So I'm gonna be quiet for 2 minutes and let you guys try to think about how you could summarize this lesson in 20 words or less. Good. So Destiny, I like that. You were here for pretty like the second part of what we talked about, so you did a great job summarizing that. Applying the different variables of an experiment to real life experiments. Awesome. Okay, so that was about 2 minutes there. If you're not quite done with your summary, that's fine, keep working. What you guys are gonna do here to demonstrate that you have an understanding of this and also that you watched or came to this lesson is we are going to have a turn into Canvas, another experiment example, same exact concept for what we did as a group. So Kim decides to study the relationship between heat and surface tension. He is going to be heating up water to 4 temperatures and then placing square wood chips on top of the water. He is counting how many of the wood chips it takes to break the surface tension. Okay. So for Kim's experiment, you're going to identify the independent variable, dependent variable, control variables, and whether or not a control group exists. So what I've done is in Canvas, in modules, it should show up on your calendar as well. It might not. I'm not exactly sure how it works, but I put here a, where did I put it is the question. Oh, I think I put it in virtual sessions. Here we go. Okay, so at the very bottom of modules, there is a virtual sessions, labs, and collaboration project module. So far, there was only collaboration options in there. Well, now, virtual session 1 exit slip is there. If you go in there, there's the PowerPoint that we went through today, right? I will also upload the video recording. And then there's the exit slip printed again. And I actually made it so that you can't access it until 10 a.m. because that's when the session is supposed to end. We'll probably end a few minutes earlier than that. So at 10 a.m., you should be able to go in and submit this. Now, what was I going to say about this? Oh, yeah. Sometimes we will actually be working on assignments that are already in Canvas. So, for example, sometimes we will do a lesson and we'll actually then do the quiz or the assignment that's related to that lesson. And then sometimes we'll have these exit slips. So the Cornell notes, what's going to happen is in the future here, on September 19th, which should be next week, right? It says Foundations of Biology Note Check. So that's from the first module. At 9 a.m., I'm going to post in here which lesson I want to see notes for. And I already told you guys a big hint, and it's this one, Properties of Water, is what I'm going to be looking for this time, right? And I want to see Cornell notes for that. Now you're going to have 25 hours to submit your notes. So in the off chance that you somehow lose your notes or you forgot to take them on that lesson or something like that, you'll have time to quickly do them. I'm not going to accept late notes, right? If you can't get your notes together in 25 hours, If you have a specific issue like you're going out of town or, you know, on that day specifically you're not able to access Canvas or something like that, then just send me an email and we can arrange separately. I'll probably have you turn in different notes. But that's where you will turn your notes in. So those are always before the final exam for that module. So that way you'll have at least one set of notes for your exam. And remember, you can use notes on your exams in this class, right? So it's really helpful to actually have notes and be able to quickly look through them and find information. So yeah, you can turn it in already if you'd like into the Foundations of Biology note check, and then that's one less thing that you have to do next week. Otherwise, on this day, I'll also just write on here Properties of Water, and you could turn it in on the 19th as well. Up to you. So, last thing I wanted to say before we're going to end the session, and then we still have about 15 minutes, so I'm going to stay around if you have questions about specific things in the class. Control group, good question from Harrison. That is the normal group or the group that you're comparing to. So a really easy example would be if you're watering plants and you're watering with water, you're watering with Gatorade, and you're watering with soda. Water is going to be our control group because that's what plants normally get, right? They don't usually get watered with soda or Gatorade. That's what we're kind of seeing if it affects it. Where can you find the slideshow? You can find it in modules. Very last module called Virtual Sessions, Labs, and Collaboration Project. And in Virtual Session 1 exit slip, you will see the PowerPoint. I'll also probably make a separate page that's called Virtual Session 1 with the PowerPoint as well. So, yeah, so what was I going to say? I'm losing my train of thought here, guys. Too early in the morning for me. Okay. So on the main page, right, you're going to see some really important information. Hopefully you've already looked through this information, but you'll find information about your collaboration projects. You might already want to start thinking about which one you want to do. There's a little tidbit here about the Cornell notes, which we just talked about. Exams, right, you can do them at home. You can use your notes. I'm really good at finding plagiarism. So if you plagiarize, I will probably be able to tell. And if I find plagiarism, you get a 0, right? The first time it happens, you're going to have an opportunity to apologize for plagiarizing, and get to make it up. Okay, Jordan, give me 1 second. Let me see if I didn't publish it. And then the segment exam, there's no retakes on that. So once you've taken it, it's done. Other quizzes and tests, you can retake them if you show me notes. So if you don't like your score, just send me an email. Hey, Miss Jamie, I want to retake this quiz. Here's my notes. And then I'll give you another take of the quiz. Our virtual sessions, we're here, right? We're going to be on the same link every week, 9 to 10. I do have a virtual session from 10 to 11 and from 11 to 12 as well, right? So if you want to meet additionally to that, Fridays are probably not the best days, but just email me and we can find a time to meet. We're not going to do a lesson every single week. We will do some lessons, but we'll also do some office hours type virtual sessions where literally I'm going to ask you guys a question, right? We're going to go over it, and then I'm just going to take questions from you. So those kinds of sessions will probably only be like 25 to 30 minutes. The lesson ones will probably be longer. Next week is our first lab, Whose Doorknob is the Dirtiest. If you can make it to MIWA, it's highly encouraged to be there in person, right? Because we're going to make the Petri dishes with the agar, and we're going to work on it. No, Harrison, there's actually like 50-something people in this class, so a lot of people did not show up today, which is not good. So Whose Doorknob is the Dirtiest? That's going to be next Friday at 9 a.m. If you can come in person, I highly recommend it. If you can't, that's fine. There's going to be a virtual session, right? And you're going to be watching us work on the lab and getting to participate from home. This lab can also be done at home. So if I didn't link the lab in here, that seems weird. I thought I did link it. I'll fix that. You can do this lab at home. So if you would like to do the Whose Doorknob is the Dirtiest lab at home, I'm going to make sure that the lab sheet is there so that you can actually work on it. Let's see. Virtual sessions and collaboration. Oh, Jordan, thank you so much for pointing out that you don't have that module. Check now. I didn't publish it, apparently. But I don't want you guys to see all the stuff from last year. Oops. I'm having way less tech problems with Canvas, though. I'm very happy. Okay. Okay. So there you go. Now you should be able to see collaboration options and virtual session 1 exit slip. Great. Okay. So at this point in time, if you don't have any more questions, you feel good to go, we're done, right? You're welcome to leave. If you'd like to stick around because you have a specific question, you want to go over something, one of the assignments, or you have a question about something else, then stick around. And please type your question in the chat for me so that I know what we should talk about. Here's the Whose Doorknob document, by the way, in case anybody wants to take a look at that. Okay, so the exit pass will be available at 10, so in 10 minutes, yes. You could already, in fact, I'll just put the question up while people are getting their questions ready. You could already be working on it right now if you want, and just have it ready to go. If you take notes on paper, just send me a picture of them. So you could take a picture of your phone or with your computer, just hold them up. As long as I can see what they say, that's totally fine. Elizabeth, do you want to go over the questions from Earth's early atmosphere? Okay. See, look, I was ready. I had to open. Okay, so I know some other people might be confused on these too. This assignment, right, it was due yesterday, I believe. So you had to compare the number of differences in amino acid sequence. Now, first of all, do we know what amino acids are? So I just sent you a little chain of kind of how this works. So first we have our DNA, right, which basically is inherited from your parents. DNA gets transcribed to RNA. And RNA basically is just carrying the information around in the cell. RNA gets translated into amino acids, which get made into a protein. So amino acids are the building blocks of a protein. It's basically like if you take Legos and you build a castle, the amino acids are the Legos and the castle is the protein. The slideshow is in the virtual sessions section and modules, yes, under the exit slip link. So amino acids differences, right, is going to be important because basically, if you think about it, if you're trying to build a castle and you have one different Lego, you might have problems doing it. And that's kind of how proteins work. If one amino acid is different, the protein might not work, right, or it might be different, or it might not matter. Sometimes it doesn't matter. So a lot of times when we see these differences in DNA, especially between humans, it's something that we call junk DNA that's not really used. No one cares about it. So sometimes the differences don't matter. Sometimes they do, right? So let's look at these questions and come to some conclusions here. So what does it mean when scientists say that living organisms share a universal genetic code? So we talked about this a little bit in the warm-up, right? To reiterate what was already suggested and said, right? We all have the same molecules that make up our DNA, and all organisms share some DNA in common, right? So even with bacteria, you do share some genes. Question 2, how does a universal genetic code? Yes, it's only available at 10, Jordan. So in 6 minutes, you should see it. Sorry about that. I didn't know we would end 10 minutes before 10. So how does a universal genetic code relate to the hypotheses about the origin of life on Earth? Isabella did a great job telling us in the warm-up that it supports the RNA world hypothesis, right? Because if we all have similar DNA and we all have RNA, right, then it makes sense that the RNA hypothesis is going to be supported by DNA. Because RNA is really the molecule that's sort of making the DNA into proteins. So how are self-replicating molecules such as RNA molecules in the RNA world hypothesis essential to the most popular hypothesis about the origin of life on Earth? So this question is kind of hard. Why is a self-replicating molecule important? Yeah, the third one I think is the hardest one, Elizabeth, I would agree. So how are self-replicating molecules important? What does that mean, first of all, if something can self-replicate? Make a copy of itself. Okay, so how are making copies of itself important? What is the RNA world hypothesis? Who could kind of summarize that for us, maybe in like a sentence or a key piece from it? Yeah, RNA copies itself to create living organisms. Yeah, basically that since RNA can copy itself over and over and over, right, it kind of has the potential to spread, right? And there's also a lot of potential for error there. There could be mistakes when it's copying itself that could have actually led to life, right? So the frequency of it being copied and also the possibility for error means that one little error could have been a result in something that could be living. Okay. Yeah, so on this assignment, guys, really the most important questions are 4 and 5. Actually looking at the genetic code and analyzing the differences, right? So if you felt confident on questions 4 and 5, amazing. Questions 1, 2, and 3 are kind of, they're a little tricky, right? And they're really not so important that you can feel like you're a pro on them, right? Really, I would like for you to know what a universal genetic code is and to know what the RNA world hypothesis is. Otherwise, you really don't need to know too much detail about these, so just do the best you can on answering them, right? Just do enough to show me that you read the lesson and clearly you are thinking about it and attempting. This kind of stuff is going to become a lot more clear later on, and in the second semester, we'll do a lab where we will translate RNA into proteins, right? And we'll kind of work on this process a little bit more. Okay, so I hate to kick you guys out. But now I am going to have a chemistry virtual session about energy, which is going to be exciting. So thank you all for attending. And I will talk to you guys soon. And I'll see you next week for whose doorknob is the dirtiest.